Friends, welcome to Biology Made Easy. Today in this video, we will discuss the fluid mosaic model of cell membrane. Earlier we have discussed that the trilaminar model is of three types. One is sandwich model given by Daniel Anderson. The second one is unit membrane model given by Robertson. But both of these models are rejected because they do not satisfy all the functions of the cell membrane. So the third model, that is also within the trilaminar model, this is known as fluid mosaic model. Fluid mosaic model, and this model was given by Singer and Nicholson in 1972, and both of them were working in the University of California. So this model is completely different from that of the earlier two models, sandwich and the unit membrane of Robertson. According to the earlier models, the plasma membrane was solid, but according to this model, fluid mosaic model of Singer Nicholson, it is a fluid structure, quasi-fluid or semi-fluid structure. That means it is not fully solid, nor it is fully liquid. It is a quasi-fluid. It is a quasi-fluid, or we can tell it is a semi-fluid. And its viscosity is almost equal to that of the olive oil. So it is that is quasi-fluid or semi-fluid. This is one difference. The second difference is the earlier models, they have suggested that both the lipid bilayer as well as the protein, they are continuous. But here again it is different. The bilayer of lipids is continuous, but not the proteins. The proteins are present here and there in a mosaic manner. That is why the word mosaic is there. That means it is a quasi-fluid and it is mosaic. That is why the name of the model, that is a fluid mosaic model. So according to this fluid mosaic model, there is a bilayer of phospholipids. So if you draw the diagram, how it is done. This is the bilayer, and these are the phospholipids. These are the phospholipids. Each phospholipid has one hydrophilic head and the hydrophobic tails. So similarly, in this side also, these are also there. This is a bilayer of phospholipids. Now, in the bilayer of phospholipid, you see these are the heads, and these are the heads. The heads are polar. We can tell that they are polar heads, and these are hydrophilic. While these are the non-polar tails, and these are hydrophobic. That means the non-polar tails, they face each other. That means they are away from the aqueous medium. That is why they are well protected. And these non-polar tails, they are towards each side, and they are attached by means of the different types of that is weak hydrophobic bonds. So this forms a continuous bilayer of phospholipids, which is a quasi-fluid structure, just like that of the viscosity of the olive oil. Now here the proteins are present, and the proteins are of two types. Those which are present on the external and internal side of the cell membrane, they are known as extrinsic or peripheral proteins. So suppose here one protein is there like this. So this protein completely spans on both sides. Similarly, there may be another protein which is partially embedded. This is partially embedded. And here there may be one that is fully embedded. It is fully embedded. This is partially embedded. So these are the different types of, these are integral proteins. These are integral proteins, or you can tell them they are extrinsic proteins. These integral proteins are insoluble and they are not easily separable. If we have to separate them, then the entire cell membrane will be disrupted. And that happens when we give detergents. The membrane is disrupted and these proteins come out. Then, here and there will be some external proteins or extrinsic proteins. These are extrinsic proteins, and these extrinsic proteins, they are more on the inner side and less on the outer side. And these extrinsic proteins, these are soluble and they are easily separable. So now you mark one thing. Now the bilayer of lipids, that is continuous, while the proteins which are of two types, may be integral proteins or extrinsic or external proteins, they are present here and there in a mosaic manner. So the whole thing appears as if the icebergs are floating in a sea of water. Icebergs in a sea of lipids. That means if the lipid bilayer is equal to the sea, then the proteins are present just like the icebergs. So as the icebergs float in the sea, similarly the proteins, they float in the bilayer of phospholipids. Then in addition to this, if some oligosaccharides, carbohydrate oligosaccharides will be attached to this protein, this may be branched or unbranched, then these are the oligosaccharides. These are oligosaccharides, and in this case this is protein, this is oligosaccharide, so this is known as a glycoprotein. And if the same thing will be attached to a lipid, then it is known as a glycolipid. This is glycolipid and this is a glycoprotein. In addition to this, here and there also there will be sterols present. Sterols, because the sterols, they provide stability to the membrane. So this is the structural diagram of the fluid mosaic model. Now, what are the important aspects of this model? So one thing is the lipid bilayer is continuous, but the proteins are not continuous, they are present here and there. But how these things they move? Now usually the proteins, they move laterally from one side to another. That means the proteins they move laterally. Lateral means likewise they can move in this way. And how fast they will move, that depends upon the fluidity of the bilayer of phospholipids. The more fluid it is, the more rapidly the proteins will move. That means the fluidity of the phospholipids depend upon the lateral movement of the proteins. That is a very important point. The fluidity of cell membrane can be determined by lateral movement movement of proteins. So this is how the proteins move. But how these phospholipids move? The phospholipids can move in two different ways. We will take that of the lipids. Now lipids have rotational movement. Rotational, or we can call it also transitional. So in this case, what happens? The phospholipid of one layer, it can rotate like this, and from one place it can go to another place. From one can come to this place in this way, or in this side one can move to the another place and one can come to this side. That means it is a rotational or transitional movement. That is one type of movement. Now the second movement is more important. That is known as flip-flop movement. Flip-flop movement. Now in flip-flop movement, what happens? The phospholipid of one side, it comes to the other side by rotating 180 degree. That means this phospholipid will come to this side. That means 180 degree it has to rotate. But what happens? The head is hydrophilic while the tails are hydrophobic. So when the hydrophilic head will rotate like this through the hydrophobic, so naturally the process is very slow. That means the flip-flop movement is quite slow because the hydrophilic head has to come through the hydrophobic tails. Now this that means the phospholipids have rotational or transitional movement as well as flip-flop movement, but the proteins have only lateral movement. They do not have flip-flop movement. This is very important from examination point of view. Then this bilayer of phospholipids is quasi-fluid or semi-fluid. What is the importance of this quasi-fluid nature? Now, due to the quasi-fluid nature, this cell can perform some functions. What are the functions? The cell division, cell growth, number 1 cell division, number 2 cell, number 3 secretion, number 4 endocytosis, number 5 that is intercellular junctions. That means because the cell membrane is quasi-fluid, it is semi-fluid, that is why the growth of the cell can take place. That means it can expand. When the cell divides, a single cell becomes 2 cells and in between one cell membrane is formed. That is possible because it is a quasi-fluid nature. Similarly, the secretion, excretions, they are possible. Endocytosis, when there is a phagosome, pinosomes are formed, that is also due to this quasi-fluid nature. Similarly, different types of intercellular junctions like desmosomes, tight junctions, all these or gap junctions, all they are formed possible because of this quasi-fluid nature. Now, the next thing is why this model is accepted. What is the reason behind it? It is accepted because this model satisfies all the functions of the plasma membrane, like the cell growth, cell division, secretion. That means all these functions are possible because it is a quasi-fluid nature. Second, by means of different modern techniques, it has been found that the proteins are not continuous, they are present here and there. Besides, this model satisfies that the cell membrane can break easily. It can break easily because it is quasi-fluid. It can repair easily, and it is dynamic in nature. That means the plasma membrane or cell membrane is dynamic, it can break easily, it can repair easily, and it can give all these types of functions because it is a quasi-fluid nature. That is why the fluid mosaic model of Singer-Nicholson is accepted at present. So I hope now you have a very clear idea about this fluid mosaic model, and this model is at present it is the accepted model. So thank you very much for watching the video. God bless you. Wish you a very bright future.